Today, we focus on brain tumors and antibody therapy.
Brain tumors.
Brain tumor is a general term for tumors that form inside the skull. The annual incidence is not high, around a few per 100,000 population, but brain tumors characteristically occur in all generations. Brain tumors arise from various tissues as well as cells of the brain itself, so they are classified accordingly into more than 100 types.
100 types.
Brain tumors vary in nature depending on type, so they are classified into 4 grades according to malignancy. More than half of all brain tumors are grade 1 benign tumors. As they are slow-growing and do not invade the surrounding normal tissue, complete healing is quite possible with surgery. Brain tumors from grade 2 onwards progressively develop the characteristics seen in common cancers. Among the most malignant grade 4 brain tumors, glioblastoma is the most dangerous one. Glioblastoma is one of a group of brain tumors called gliomas. Brains are composed of nerve cells, or neurons, and glial cells. 4 types of glial cells fill the gaps between neurons, and play various roles in assisting the function of neurons. Brain tumors arising from these glial cells are called gliomas. Among gliomas, the treatment outcome for glioblastomas is particularly low, and even among all cancers, glioblastoma is as lethal as pancreatic cancer.
Is there any treatment available?
As the growth is very fast, surgery should be done immediately, but due to invasion, it is pretty difficult to remove it thoroughly, considering the damage to brain function. Then, radiation and anti-cancer drugs are used to slow the growth of the remaining tumor. These are the standard treatment, and unfortunately, we haven't found anything better at present.
How about antibody treatment for glioblastomas?
Therefore, the Department of Antibody Drug Development focused on the protein called Podoplanin.
Podoplanin.
Podoplanin is a membrane protein found in kidney podocytes. In addition to podocytes, this protein is also present in the lungs and lymphatic vessels, and is necessary for lung and lymphatic development during fetal life.
Then, this is also an important protein.
Podoplanin is found to be increased in various cancers, including brain tumors, and correlates with malignancy. Podoplanin is characterized by the PLAG sequences, repeated in its extracellular region. The PLAG sequences interact with the CLEC2 protein on the surface of platelets, and induce platelet aggregation. This function is important for the role in normal cells, introduced earlier, but cancer cells with increased podoplanin make bad use of this function to facilitate metastasis through blood vessels.
Platelets are what stop bleeding in wounds. What would cancer cells do with such things?
There are several obstacles to cancer cells metastasizing through blood vessels. One is the shearing force generated by the blood flow. Most cancer cells that enter the blood vessels are physically destroyed. Another is attacks from immune cells that circulate in the blood.
I see.
But if cancer cells succeed in aggregating platelets on their surface using podoplanin, it will provide a cushion to soften shearing forces and a cover to escape immune cells, and help cancer cells survive in blood vessels. In addition, cytokines and growth factors released from platelets also aid metastasis, by suppressing the activity of immune cells and promoting the extravasation of cancer cells.
They do outrageous things.
This is why the amount of podoplanin in cancer cells correlates with malignancy.
Then, with podplanin antibodies, we can attack cancer cells and prevent them from metastasis.
Right, it can be effective against podoplanin-positive cancers. The Department of Antibody Drug Development has produced a number of anti-podoplanin antibodies, and registered them in its antibody bank. It has been shown that these antibodies have an inhibitory effect on cancer growth and metastasis.
Amazing.
Brain tumors rarely metastasize, but we can expect anti-tumor effects on podoplanin-positive ones. However, previous antibodies reacted with podoplanin on normal cells as well as those on cancer cells. So, cancer-specific antibodies, CasMabs, were developed to make it safer.
Does the fact that CasMabs were established mean podoplanin differs between normal and cancer cells?
One of the features of podoplanin is its many glycosylation modifications. It seems that podoplanin on cancer cells has unique glycosylation that isn't found in podoplanin on normal cells. Some of the several anti-podoplanin CasMabs are thought to recognize such cancer-specific glycosylation. Others are considered to recognize local conformational changes caused by the cancer-specific glycosylation, rather than the modification itself.
I see.
The research investigated the cytotoxicity-inducing effect of an anti-podoplanin CasMab on glioblastoma. Like the podocarlicin CasMab presented in video number 5, this antibody has been modified to improve its safety as a drug.
It's antibody humanization. This makes the antibody less foreign to the human body. Then, defucosylation increases the ADCC activity.
First, the binding of antibodies to cells was examined by flow cytometry. On the left is the result of hamster cells without podoplanin. And on the right is the result of hamster cells expressing human podoplanin.
The red peak of CasMab is on the right, when the cells have podoplanin. It shows that CasMab bound to the cells by properly recognizing podoplanin.
And this is the result when podoplanin-positive cancer cells were used. Cells derived from brain tumors are on the left, and cells from lung cancer are on the right.
The data show that the CasMab is able to bind to cancer cells.
Next, the cancer specificity of the CasMab was tested by immunostaining. The previous antibody bound to not only cancer cells but also normal cells.
It bound to the lymph vessels.
Here's the result of the CasMab.
Wow, nearly no binding to normal lymphatic vessels. What is its capacity to induce cytotoxicity?
For more information on cytotoxicity induction by antibodies, please see video number 2. First, let's see the induction of antibody-dependent cellular cytotoxicity, ADCC. The result from brain tumor cells is on the left, and that from lung cancer cells is on the right.
I can see the CasMab effectively induced ADCC in both cell types.
And the results of inducing complement-dependent cytotoxicity, CDC.
The CasMab induced CDC too. The CasMab was able to induce both ADCC and CDC in podoplanin-positive cancer cells. Then, it likely has an anti-cancer effect.
Here are the effects on cancer growth. Brain tumor cells on the left and lung cancer cells on the right. The data showed that the CasMab treatment inhibited tumor volume growth.
We made it.
In this video, we introduced the cancer-specific antibody, CasMab, against podoplanin. Like podocalixin introduced in video number 5, podoplanin has been known to increase in various cancers and has a relationship with cancer malignancy. However, it was rarely regarded as a target for antibody therapy considering the risk of side effects due to its wide distribution in normal tissues.
But with CasMabs, it's a different story.
Right. We expect an expansion of treatment options for podoplanin-positive cancers.
I hope it will lead to an improvement in the treatment of the most malignant glioblastoma, for which we currently have no effective treatment.
The Department of Antibody Drug Development is now developing antibody-drug conjugates, ADC, and chimeric antigen receptor T cells, CAR T cells, for this antipodoplanin CasMab.
I believe that they will enable more potent attacks of cancer cells.